Hi, this is Dr. Lauren Launen, and this is the beginning of a lecture set on bacterial and archaeal genomes, in other words, prokaryotic genomes. And I want to remind my listeners and viewers that a genome is all of the DNA within an organism. And the reason I'm reminding you of that is to also remind you that the bacteria and the archaea are the two domains of life for which each organism is in fact one individual cell. These are not multicellular life forms. These are prokaryotes. They're single cellular life forms. So we're going to go through some content, and at the end of it you should be able to knowledgeably describe the basic structure of prokaryotic chromosomes, be able to describe the basic structure and function of plasmids, discuss some fundamentals of chromosome compaction, and consider the role of chromosome compaction on the biology of the organism involved. So, bacterial and archaeal chromosomes. Typically in the prokaryotic world, each organism is comprised of one cell, and that one cell has one single circular molecule of DNA or chromosome. It is found located in an area of the cell called the nucleoid. It is anchored into the nucleoid by a set of proteins that are somewhat comparable to the cytoskeleton. So, although we don't call it the cytoskeleton, it's a cytoskeletal-like structure, and it'll anchor the chromosome in place. There is no nucleus, because these are prokaryotes. And let's give you a feeling for the size of these genomes, okay? In E. coli, there are about 4,000 to maybe 4,200 genes that are spread over a total of 4.6 million base pairs of DNA in that one chromosome. And if you were to take that chromosome and you were to pull it apart out of the cell, spread it out and flatten it and tease it apart, it would be about 1.2 millimeters of DNA, which isn't a size that you can actually visualize with the naked eye. And of course, you can't see a single cell of E. coli, so how can you fit something that's big enough to see with the naked eye into a structure that's too small to see with the naked eye? And by the naked eye, I mean not assisted by a microscope. Like, how do you do that? How do you pack all of that material inside one tiny cellular space, and even within an area of that cellular space? And so the secret to that is compaction. DNA is very highly compacted in order to fit into any cell, including prokaryotic cells. And one way to visualize that, this is an electron micrograph. This is an E. coli, and it has been lysed, it's dying, it's damaged, and it is releasing its DNA out into the environment, and boom, out comes all this DNA. And see how it's all these thread-like substances? That's all the chromosome. It's being kind of torn apart and fragmented in the environment, but all of that was coiled up in the double helix and then coiled up some more and fit into the E. coli. All right, so bacterial and archaeal genomes. So here's a cartoon depicting a typical prokaryotic cell, and we just talked about the fact that each cell of a prokaryote will have one circular chromosome, and it will be anchored at an area called the nucleoid. Now, that is not the only genetic content found within prokaryotic cells. Most prokaryotic cells also have small extra chromosomal pieces of DNA that are called plasmids. And in this figure, the plasmids are drawn in blue. So we've got 1, 2, 3 plasmids. These are not chromosomes. They're molecules of DNA called plasmids. And they are extra to the chromosome, which is over here. And that's why we call them extra chromosomal. They're also, like in this figure, not really drawn to scale, in that the plasmids are usually a fair bit smaller than the chromosome. So let's think a little bit more about genome size. So this is a diagram from a text called Essential Cell Biology, and it gives us ranges of what are called the haploid genome size. Now, you should be perking up your ears right now because this is a lecture about prokaryotic genomes, and I just used the word haploid. And haploid, in fact, is a word that applies strictly to eukaryotes. Eukaryotes can have diploid genetic content in their cells, or they can have haploid genetic content in their cells. And in eukaryotes, diploid means you've got both copies, both members of the pair of homologous chromosomes within the genome found in that one cell. And the term haploid means you're looking at a cell that's got one copy of each chromosome. And that in humans, for example, would be only in our sperm or our eggs. And of course, prokaryotes don't make sperm or eggs. Prokaryotes always only have one copy of their one chromosome. And so we make graphs like this. We try to normalize the data. And so down here we're comparing prokaryotic organisms, the bacteria and the archaea. We're comparing their genome size against eukaryotic organisms, which are from here all the way up here. There's humans right there. So that we can compare apples to apples, we're documenting only the haploid genome size. Note that this is a log scale here, okay? So the E. coli genome, which as I mentioned was about 4.6 million base pairs, it's shown here. This is a log scale, 10 to the 6 is a million, right? This would be 10 million, so some bacterial genomes are in the 10 million range. Most are sort of in the million base pair range, okay? And the archaea a little bit smaller, okay? So the mammals tend to have a lot more DNA, so they get up to, billions of base pairs of DNA. There's the human genome, it's in the billions, okay? Not the tens of billions, but the billions. Birds a little bit smaller. Amphibians are quite wide-ranging, as are the fishes. The plants are really wide-ranging, and interestingly, so are the protozoa. They have widely diverse genome sizes, even though they are single-celled critters. So bacterial genomes tend to be much smaller on average than eukaryotic genomes. That's the take-home message from this slide. And again, considering that genome size aspect, here's some more information in that regard. So up here is Homo sapiens, that's us, right? We've got about 3.2 billion base pairs in our genome. We have around 25,000 genes, and the average gene density, like number of genes per nucleotides or per bases or base pairs of our genome, is 1 gene per 100,000. We have 46 total chromosomes. Okay, so let's take a look down here at the E. coli genome. So it's about 4.6 million in size. There are about 3,200 genes, okay? And there are, on average, there is a gene density of about 1 gene per 1,400 bases. So the gene density is higher in prokaryotes than in eukaryotes on average, okay? So that means that most of a prokaryotic genome is coding, whereas a lot of a eukaryotic genome is non-coding, much of which is regulatory or repetitive sequence, which we have not yet figured out the function for. Okay, so let's think a little bit more about this idea of compaction. How do we fit all of the prokaryotic DNA that these organisms have into a tiny area, into the location called the nucleoid? Okay, so the nucleoid is going to take up about 1/3 of the volume of the cell, right? And the secret to fitting a lot of DNA into a tiny, tiny volume, all right, so let's look at E. coli again as an example. Here in the red box are 2 different bacterial species, E. coli and Haemophilus influenzae, which causes a flu-like illness, as you could have probably guessed from the name. They have DNA genomes. Their DNA is double-stranded. And if you were to take the DNA and pull it apart, these units are in micrometers, but 1,200 micrometers is about 1.2, or is exactly 1.2 millimeters long. And the size of an E. coli is about 2 by 0.5 micrometers, the head of it, okay? So this is a really, really tiny area, right? It's a tiny, tiny area in which to stuff all of this DNA. And how do you do that? Well, you do that with the help of proteins. So there are a lot of proteins that are referred to as non-histone proteins. We'll learn about histone proteins when we look at eukaryotic genomes. So non-histone proteins help bacteria and archaea to fit DNA into the cells in a process called compaction. Okay? All right, there's another trick, okay? So this is an image of an E. coli chromosome taken or drawn at different stages of growth. During exponential phase of growth, when the E. coli cells are extremely active and they're doing a ton of metabolism, it looks like this. During stationary growth, when they're just kind of sitting around waiting to do something, they look like this. So these flower-like patterns are how we see the chromosome when it's actually at the nucleoid region in an E. coli cell. And this is true for all bacteria and all archaea. And these flower-like structures, each area is called a looped domain. The formation of looped domains is a way to pack DNA. And so one of the mechanisms whereby prokaryotic cells fit DNA into the tiny spaces required is they use something called loop domain formation, and that looped domain formation relies on non-histone proteins. Okay? There's something else. There's a second factor here. So the second aspect of fitting or compacting prokaryotic DNA into tiny cells is to form structures that are called supercoils, or to do what is called supercoiling. And that's an extra level of twisting beyond the formation of the double helix that is imposed on prokaryotic chromosomes in order to fit them into the cell. So if this is in a relaxed form a prokaryotic chromosome, each red line that we see here, this red line that we see here, there's a double helix in there. So there's already a level of twisting, right, in this entire structure. Now if you take the whole thing and you twist it again, note that this twisting pattern is different from this twisting pattern, right? Here this strand is on top, over here this strand is on top. Everybody good with that? This pattern is called positive supercoiling. This pattern is called negative supercoiling. And you have to look at this area in order to determine whether it's positive or negative. All right, so what affects supercoiling? Supercoiling is not static, nor are the looped domains themselves static. So the architecture or topology of a chromosome is actually constantly shifting at local areas and in totality according to what the cell is doing. In this image, we're seeing the role of transcription and we're seeing the role of DNA synthesis, which are two cellular functions that can affect supercoiling. Transcription, when you should know what this protein is, right? What is the enzyme that does transcription? It is RNA polymerase. So here's RNA polymerase and it's traveling along and it is transcribing one of these two strands. I can't tell from this picture if it's using the green or the red as the template, but I can tell you this is the new RNA being formed and one of these strands is the template and it is heading that way. And you can note that there is positive supercoiling happening in front of the RNA polymerase and negative behind. And the biophysicists figured this out for us. So the act of transcription changes the pattern. Similarly, in DNA synthesis, you'll get positive supercoiling in front of the enzyme DNA polymerase, and you'll just see relaxation of the supercoiling structure behind it. Right, I'm going to close this and move on to the next part of this video lecture set.